There we go. Perfect. All right. Welcome, everyone, to our April MLX forum. Today's theme is going to be computer vision focus. We're going to have two speakers, one in healthcare and the other in agriculture. So the first speaker will be Matthew Blomquist talking about ultrasound and how to measure knee kinematics. And Will Delab, sorry, I messed it up now. De La Bretonne. Okay, sorry about that. De La Bretonne. I should say it 3 times. Plant breeding, I believe using drones to analyze how plants are growing. So these are going to be, I think, pretty interesting talks. I'm so excited to hear about them. Moving on to our next slide. It's not advancing. Chris, what did I do to it?
I don't have a touchpad.
Okay. Click there. Oh, okay. Perfect. So we want to thank our sponsors. Without this, we wouldn't be able to feed you lunch. So let's note that Epic and Promega and Wharf help support our initiative here. One quick note is that we have ML Plus coffee tomorrow. And so this is a little bit more casual. Basically an opportunity to get free coffee, but also socialize and discuss some challenges. Tomorrow we'll be talking about maybe the opportunity for ML methods to help process data for this neighborhood study to actually look at health disparity research. And that's tomorrow here on the first floor at between 9 and 11 a.m. And one more final note before we get started, we're looking for our May presenters. So if you're either online or in person and know of somebody or would like to present for our May forum, please consider. And you can sign up with that link on the form. And also you could reach out to myself or Chris Endeman, and we'd be happy to make the connection for you. So without further ado, let's bring Matthew up and he'll begin his talk.
All right. Thank you very much for having me. I will be presenting my research titled an ultrasound-based method to measure knee kinematics enabled by deep learning. So first of all, I want to say thank you for letting me present and speak about my research today. Thank you to my lab, the BAM lab, my advisor, Josh Roth, and especially Chris. I think I came to Chris about maybe November of last year, and I think I knew what machine learning was, and that was about it. Not anything you could do with it, so he's helped me immensely over the past months. Just a quick breakdown of what I'll be going through today. So start with some background information just to provide some context. Hopefully you can follow along a little bit better. So my lab studies knee biomechanics. So one of the kind of fundamental things that, especially for knee mechanics, is called the laxity test. And there's a bunch of different laxity tests, but basically a laxity test determines the passive stability of a joint. So this is done to test for excessive looseness in the joint. So if you look at this image, the solid black line right down the middle might be like the neutral position. And then if like a clinician or a doctor, someone applies a load to the joint, they keep the femur or your thigh steady and they move your shank or your foot or your tibia. If there's a rotation there, that's what the laxity is. It's a difference in the angle or translation or whatever it may be of a joint. And this is done for a variety of reasons. It's usually done to check if like the integrity of tissues beneath the skin. So, I mean, a common example is an ACL tear. Athletes have ACL tears all the time. So they might do a laxity test to see, okay, is this ACL there? Is it working properly or not? So it's very common and very important to be able to measure what the laxity is in different joints. And so the 4 main ones I'll be speaking of and are of the knee. And I'll be speaking of anterior, posterior, varus, and valgus, and that's just different directions. So anterior is the top left image here, and that's keeping your thigh in the same spot and then moving your calf forward. And then posterior is the opposite. You move your calf backwards. And then varus is like bow-leggedness. You keep your thigh in the same position and then move your foot inward. And then valgus is like a knee knocked, I think is what they call it. And you move your calf outwards. So those are the four main ones that I'll be talking about in testing today, just to provide some background. So current methods that groups use, like in the clinical setting to measure laxity, are just they're subjective. The doctor's done it many times, so they can do it by hand, and this works when you have a big difference between things or between movements. Like if you have an ACL or if you don't have an ACL, usually they can tell those. But there's definitely limitations in the subjectiveness of this, is they're not accurate to small differences. Maybe you have some tissue in your knee that's partially torn but not totally torn, and they can't tell the difference between a healthy or versus not healthy. It's also not dynamic. You can do this in a static pose, but you can't check what the laxity is, how much your joints are moving around during like walking or whatever it may be. There's also different like stress radiographs, which are really good. Basically shooting x-rays into the knee you can do it pre and post a load or before and after applying a load. And these are also really good, but the limitations are they have ionizing radiation. So that just limits like how much or how often you can do this. And then also they're not dynamic, just as before. They have to be in a static pose. So kind of what my research is focused on is ultrasound. Ultrasound may be a safe alternative to quantified joint motion. And so we know ultrasound is safe, we know it's accessible and broadly familiar to both clinicians and researchers. So it's pretty translatable, but we don't know if to measure bone motion, if it's accurate, precise, or we can use it in dynamic tasks. So the last thing I want to go over is I'll show some images of ultrasound. So I just want to go through kind of what we're looking at when we look at an ultrasound image. So this is my creation of a cartoon knee right here with an ultrasound transducer on the side of it. And so ultrasound sends sound waves, just like it sounds like, sends sound waves, and these sound waves reflect off different tissues or bones within the knee. So if we look at an ultrasound image, this is an actual one that I captured. If we look at this ultrasound image, you kind of look at it from the outside looking in. So the right is the outside and the left is looking in. So the right is the skin surface, and then all the white material that pops up, that's all the tissues reflecting the sound waves. So the kind of rightmost layer is different, like maybe ligaments or tendons. And then the two broad bands right in the middle of the image are bones. So that's what we're interested in seeing. We're interested in seeing how your thigh bone or your femur and your calf bone, your tibia, how they move relative to one another. So data collection, what I've collected so far, I want to have a warning here. I have a picture of a cadaver knee on the next two slides. It looks exactly like this, but obviously not cartoon. So don't look if you don't want to see that, but I'll let you know when the slides are over. So for in our lab, we have a big 6 degree of freedom robotic testing system, and 6 degree of freedom means that it can move in any translation or any rotation in the workspace. So right around there. So we can prescribe like cadaver knees doing walking or different laxity tests or however it may be. So it's a pretty cool tool that we have at our disposal. And you can input loads or motions. So you can say, okay, go to this position, or you can say, okay, apply this load. So it's really nice to be able to do like laxity tests that I said before. Okay, apply this load to the knee in this direction, like this. So you have one primary load, and then say all other loads should be 0. And then it can also output the loads or motion. So if you prescribe this load, okay, it tells you this is how much it actually deflected in this area, or how much it translated in both the primary direction that you prescribed and also the other directions. So it's pretty cool tool that we have. And for the testing that we've done, we have an ultrasound transducer that we attach, or we put on the knee to image the bones. So I guess you can't see my mouse, but so this transducer right in the middle picture, we place right over the knee. So it's a pretty low profile ultrasound transducer that's pretty convenient to measure bone motion. And so what I've tested so far is the 4 laxity tests that I talked about before: anterior, posterior, varus, and valgus. And we've tested so far 5 different specimens and 6 different tests per direction. So like 6 for anterior, 6 for posterior, and so on. And so for each direction, we have 30 total tests. All right, this is my reminder to say that cadaver pictures are done now. And so this is just an example of one of the tests. So the test is going from unload to load to unload. So if you look at these cartoon images, if you look at the bone or the space between the bones, they should open up. And so if you look at this image on the right side, you'd see it open up and then come back together slightly. So what we want to do, we'll play it for 1 or 2 more cycles. So what we want to do is, if we give some machine learning model these ultrasound frames, these videos, and it track how these bones, how this like white prominence in the ultrasound images, how the bones move from frame to frame. Or in other words, if we input like 2 consecutive or sequential images, feed it into some machine learning model, can it tell us what the transform, what the translations and rotations between these 2 images are? So that comes in, or this is where machine learning, deep learning come in. So like I said before, this is my first like machine learning or deep learning project. So right now I'm using PyTorch, and we're using convolutional neural networks. So from my understanding, convolutional neural networks are very good at condensing images with tons of data to be able to process more efficiently. Thus, they're very good at learning important spatial features. And so I found a paper from 2018 that also used something similar to this, that put in pairs of ultrasound images. And so I've been like basing my architecture based on this, but all these ultrasound images that we use are grayscale. So basically you pair the 2 frames that you want to tell what the motion between the bones are. You pair those as a multi-channel image, and then it goes through just standard convolutional activation and pooling layers to a fully connected layer. And then this is 6 because they did 6 translations and rotations, but mine is actually 1. Our data set, like I talked about before, we have 5 specimens worth of tests, 6 tests per specimen per direction. So that's 30 total tests per direction. And so what I've been using this for is 1 specimen I've kept out for the test data set, another 4 are the train data set. And right now I've been using a 90-10 split for training and validation. And in total, this comes out to a little under 5,000 frame pairs, which ideally we would have more. So one thing that we've been working on is augmenting the data as well. So we use three main techniques to add more data to our data set for augmenting the data set we have already. So the first one is adding blur. Hopefully you can kind of see that, but the top 2 images are the original ultrasound images, and then the bottom 2 we add a blurring technique to. And then we also add random rotation. So anywhere from -10 degrees to 10 degrees, we rotate the images. And then we also add noise to the images. So we essentially double our data set to get about 9,500 frame pairs. Then more of a recent change, we've also added in a long short-term memory layer, so LSTM layer. So convolutional neural networks are really good at feature learning. Long short-term memory are good at sequence learning. And I had no idea what this was before. So if you don't know, LSTMs are kind of a recurrent neural network designed to process sequential data that have long-term dependencies. So this is kind of the overall structure that we have. I guess some of these terms I haven't mentioned before, but basically we input this cine loop, which is the ultrasound video of the whole trial. It goes into our convolutional layers, and it goes into our LSTM layers, and then it goes to the fully connected layer. And finally, the output is the relative motion between the bones of the input. So preliminarily, this is a plot of the y-axis is the motion. So in this case, it's a rotation. The x-axis is the frame number. So basically, you can think of it like time. The black is the robot, which is our gold standard, what we're trying to compare to. And so it goes up and goes down. And a red is our prediction. That's the ultrasound predicted kinematics. So this is the case with the convolutional layers, but no LSTM layer. as you can see, it goes up, but it just keeps going up. So it doesn't track very well. So we're kind of like, what the heck's going on there? And so that's when we kind of implemented the LSTM. And this is a good case of the LSTM, but it tracks very well. It follows the curve very well. A little higher, but overall, we're very pleased with that result. But they're not all perfect. So this is a CNN LSTM that's not so good. Overall, we're happy that they all kind of follow the trend of going up and going back down, but this one just really underpredicted how much rotation was in this trial. So our overall takeaway is the results are promising and they're good, but not great. So finally, I just want to kind of go over some challenges. The first one I'm sure everyone kind of has is the just computation resources. And they're just, right now we're limited in what we can use. So what I've been doing is a cross-validation. So if we have 5 different folds for the 5 different specimens, we use one specimen for a test and then train on the other 4 data sets. And so if we want to do a complete cross-validation that requires 5 different models that we create, right now I've been doing everything locally on my local computer. And so my kind of method to speed that up was to use other computers on my lab floor in Wimmer to get more access to use more computers to run more models. So it's the short-term use. Those computers are actually gone now because someone's moving into that space, so I can't do that anymore. But the long-term kind of potential solution is to work with CHKC to set up running more through the high-throughput grid. And then also limited memory on the computers has been something that I've been running into. So kind of my solution is just to downsample the data or downscale the data. And so far it's worked, but I don't know how much I'm kind of missing out when I downscale the data. So that's kind of a challenge I've been running into. Also limited data set. Like I said, we only have 5 knees so far. We actually have a couple more, but I haven't processed the data yet. So the challenges with this is just the cost, time, and resources we have. So the cost, it can be, I think it varies a little bit, but some like ballpark, like $500 a knee. So it's not cheap to get a cadaver knee. The time it takes is 1 to 2 days to dissect, prepare, and then complete the testing. And then also resources, I should put ethics of it is we want to get as much use because these people donated their bodies to research. So we want to get as much use out of it and get any use that we can possibly get. So kind of our potential solutions that we've worked with so far is if we can work with other lab members in my group to see if we can use a knee for potentially other projects, and that kind of spreads out the cost across multiple projects. Multiple people can help dissection and testing and preparation, and then also just you feel better about putting the knee to more use, getting more out of it. So another challenge is just the network model and architecture. So kind of the first big question is the CNN or CNN plus LSTM the best model to use for this application? And if it is, kind of what parameters to choose, like how many layers should I do? Should I add dropout layers? Are there other features like the LSTM that could help the model? So that's kind of something that I'm sure everyone runs into. That's pretty application specific. And so my potential solution right now is, I don't really know, but I'm just kind of ongoing work is to test out all these parameters and kind of see what works out best for my application, and also talk with others that kind of do similar stuff to see what they suggest. Then finally, transfer learning has been a little bit of a challenge. We want to see if we can use a pre-trained model. So far, we have been using ResNet, and ResNet hasn't been too successful. We just think that the images that ResNet's been trained on is just very significantly different than ultrasound images. So it hasn't been going too well with that. But our potential solution is just to try other pre-trained models to see if we can use a pre-trained model to get more data and get better accurate models. So finally, just the questions and discussion. So I have some questions or discussion points on here. in regards to kind of the challenges that I've been working towards. but also I'm happy to take any questions or discuss anything with the audience. And we do have a question on chat that says, is the age of the deceased who donated the knee known? Yeah, we do know the age, the height and weight, and therefore the BMI. So we do know those things from the donation. So that is helpful in case we want to look at specific groups, like if there's a diseased knee or if we want to look at only healthy knees. But kind of my application is I want to look at that, like the all encompass, can this be used for all these different applications? But yeah, thank you very much for listening. I'm happy to talk about anything.
Curious if you looked into any potential like ultrasound phantoms.
Yeah, there are. We've done a little bit. There's a lot of things online that like simulate ultrasound images to get more data. I think that's a little bit of at least what we start with, that's a little bit of a learning curve. So I think that's definitely what we want to see if that's an option. The other thing that we talked about is to see if we can kind of, there's different groups like chemists that have big databases of like scans of ultrasound knees. So if we can use some like self-supervised model to like try to pick out what the curves look like in all those, and then we can like use transfer learning to apply it to what we have, like actual target data and stuff like that. So that's kind of ongoing work, but yeah, we haven't looked at like actual ultrasound phantoms, I guess, but different like simulating ultrasound images for us. We have talked about a little bit.
Yeah, I mean, you might also discuss Scott Reeder, but I think he's got a ultrasound phantom company.
Oh, really?
So, and I don't know how detailed they've gotten. You might be able to have a simulated knee joint with known densities.
Okay. Scott Reeder, you said?
Yeah,
okay.
He's the chair of radiology.
Okay. Cool. Very cool.
I don't know. Did they have a knee phantom? I thought it was mostly for liver fat direction.
It might be. I don't know the range of the phantoms they have.
I believe that's primarily the focus.
Okay.
Right.
But yeah, I think pre-training would be a good idea.
Yep.
One thing I consider too is like once you have your configuration, like the apparatus set up, I assume ultrasound is sort of free at that point.
Yeah.
You can scan as long as you need to.
Yep.
So you might be able just to get like a lot of data just in that configuration. I think there's a question in the back maybe, but I have one quick question. What is the step, or how do you hope to translate this to human use in the future?
Yeah, so
steps necessary for that need?
Yeah, so I think this is specifically for laxity tests. So I think this could be used by clinicians to test for laxity. They have all these devices already, but also I think it's like the longitudinal aspect of, can we test over time? Can we test these specific groups over time? So I think, I mean, it kind of depends of what the accuracy or what the variability of our measurements can get down to. But I think the goal is to apply them in both clinical and research applications. So I think the big goal the hardware aspect is getting some ultrasound that we can secure to the knee, easily put on, take off for clinicians and researchers. And then, yeah, kind of see how accurate we can get this to see what applications it can be used for. Yep.
Have you thought of simplifying and using techniques like optical flow?
We've done that a little bit. The one issue with that is I guess it doesn't address this too much because we're doing it in 1 degree of freedom or one direction, I guess. But we do want to apply this in the future to things like walking and stuff where there are autoplane motion, which I think would, I guess, where the speckles of the ultrasound, where these individual So, yeah, I guess autoplane motion could be challenging for optical flow, just because optical flow is in the plane of it. So I think we've done some stuff. So another study we did was cross correlation. So that's just 2D. And it worked decently well. We're just trying to see if like a machine learning model would be better, I guess.
Yep. So you, I believe you said, so with the ultrasound, you only get one measurement for your time step.
Yep.
And then you make images by taking that time step and the next time step. So you have a 2-channel image essentially.
Yeah. So actually we like slap the ultrasound transducer on, and then we basically turn the ultrasound on, and it does the motion, and we track it throughout the motion. So we have like 1,000 frames for the entire motion.
Yep.
And then we input like the first and the second frame, and then the second and the third frame, and so on.
Yep. But have you tried, like, maybe one thing to help you get more data is, can you do like the first frame, the 5th frame, and the 10th frame?
Yeah, we do.
If you can get good correlation, show it like.
Yep, we do that.
Okay.
Yep. Yeah, that was a good idea. But yeah, we actually do like the first and the 40th frame, and the 10th and the 50th frame, and then 5th and 20th or whatever. Maybe that's one way that we try to amplify our data set, I guess.
Yep. We actually developed a purely 2D ultrasound for a 2D convolution for freehand ultrasound construction.
Oh yeah.
So if you're interested, like, we can check afterwards.
Yeah, that'd be great.
So we're actually testing that. I think CNN with LSTM is not a good idea to do that.
Okay.
And essentially, we're doing that. We are using physical property of ultrasound, so that we can use pure, essentially use 2D CNN central feature extraction.
Okay.
And we can have better results compared to others. And accuracy, we can talk with it.
Yeah, that'd be awesome. Thank you. Awesome.
I have a question for the room, sort of. Has anyone here tried bone AI models? Yes, is that a difficult process, or do you need to request access to the models? I assume they're trained on sensitive. Right. So I guess maybe we should try to get for the audience. The question was of the MONAI. So MONAI is a framework for deep learning and medical imaging. It's essentially like a layer built on top of PyTorch. So I only let people in my group actually use MONAI for new projects. It has a lot of like baseline models that are already created. So maybe not one exactly for your application, but I think it would be a good place to start. So I'd be happy to chat more too about our experiences with MONAI, things like that.
Are those models posted publicly typically?
They have what's called a model zoo.
Okay.
And so you can get like existing models, but then there's also like untrained, like bare bones existing models. And they have a pretty good tutorial section as well that sets and shows a lot of different examples.
Do you know if they like anonymize the data that's used?
Typically, the examples will have anonymized data from like Kaggle or the Cancer Imaging Archive or other open source applications. Yep. Yeah. Well, any other questions? All right. Let's thank the speaker. All right. Here we go. All right, so we have Will de la Bretonne. He'll be coming and talking about plant breeding in the age of computer vision. Yep. Yep. Good.
So, okay, that's it.
Yes, sweet.
Awesome. Well, thank you very much, everybody, for coming out today. And thank you, Chris and the MLX+X community for the chance to present. Really appreciate it. Before we get started, just show of hands here. Did anyone grow up on a farm? There we go. Kind of, sort of. Anyone been in a field? Any kind of agricultural field? There we go. We're in Wisconsin, right? So agriculture is my passion. It's something that I love. And also, if our farmers disappeared tomorrow, we would all become farmers, right? So it's something that we should all be very interested in. And not only how we currently do it, but how we progress it for the future. And so before I get started, I just want to say that in general, we are behind in agriculture. I mean, we're not like a couple years behind. We're like 30 years behind, guys. And so if I say stuff that doesn't make any sense, by all means, feel free to correct me. I do not know what I'm doing, but that's why I'm here. So before we get started, I have to talk a little bit about what is plant breeding. Okay, and so plant breeding is this super cool niche. It's kind of a mix between agriculture, data science, math, statistics, all that kind of stuff. But in general, it's the idea that we can selectively improve our crop varieties over time. And so just as an example of that, on the right here, we have teosinte, which is the wild progenitor species of modern corn. On the left, you have modern corn ear, right? Very different. This one has like 10 kernels. That one has tons and tons of kernels. And so the way we did this originally was entirely inadvertently. We've been doing this process for about the past 10,000 years. And so there's a lot of cool stories and history behind how this happened. For example, in corn, the Aztecs started selectively breeding because they thought there were different gods in the different colors of the corn. So to respect those gods, they planted them in different locations, right? No knowledge of how plants reproduce, that it would be any kind of benefit whatsoever. Kind of similar story for all crops across modern agriculture. And so recently we've turned to using more genomic data, more phenotypic data to help drive these selection decisions to help increase the genetic gain. So we want to continue to produce bigger and bigger corn ears. And so if this is something you're interested in at all, there's just a couple brief reading suggestions for you. So anyone been to McDonald's? Like the French fries, right? So those fries, that potato was actually developed by a guy named Luther Burbank around the turn of the century. There's a cool book, The Garden of Invention, that's kind of about his life. He was this crazy cat, moved out to California, kind of started the whole horticulture industry over there. Super, super fun book to read, not very scientific. This other guy is someone that I grew up idolizing. This is Norman Borlaug. He was given the Nobel Prize in 1970 for the Green Revolution. And so he's actually from Minnesota and spent most of his life down in Mexico developing semi-dwarf wheat and small grain varieties. So a lot of really cool history around plant breeding. It's been around forever, but the question is, how can we incorporate machine learning into that now to help push us towards the future? And so this is just a general overview of a typical breeding program. And so what we're going to do is we're going to take corn variety A. Maybe it has a really wide corn cob. We're going to take corn variety B. It has a really long corn cob. We say, hey, we want one that's wide and round, right? It's going to have more kernels per ear. We would cross those two and generate a ton of progeny. In this case, potentially millions. Typically, it's on the scale of hundreds of thousands. And the vast, vast majority of those offspring are going to be worse. They're not going to have that big, wide ear. They're just going to be worse than either of the original two parents. And so we then have to spend the next 10 years trying to dwindle down to only one of those offspring that we're going to select as a recommended variety that will then grow and release to farmers so they can grow in their fields. And so throughout this process, we have to evaluate thousands and thousands of plants every year in the field. And so currently how we do this is just by eye. So there's tons of traits that we measure, everything from leaf angle to plant height to biomass to flowering time, you name it, we try to measure it. Typically what that means is people like myself end up spending the entire summer walking around with sticks, measuring things on plants, which is great. I love doing that. I have no problem doing that. It's what I'm passionate about. And there's a lot of people like me that love doing that. But there's a fundamental limitation on how many plants you can walk to within a day, right? There's only so many of us. There's only so much ground that we can cover. And so we end up going with a lot of our plants that we put in the ground, we don't even collect data on just because we don't have the time to do it. And so currently we're in this really unique position with recent advances in drone technology. And so we think of fundamentally what we're doing when we're walking around measuring things is we're using our eyeballs, right? And we know that our eyeballs are really just these fancy RGB cameras. And so we're at the point where these drones are affordable enough that breeding programs, public breeding programs, I should say, around the country can buy these. There's commercial software available to handle a lot of the image stitching, and for the flight planning and that kind of stuff. So we can collect a lot of image data on these plants. But unfortunately, we are all agronomists and we collect this data. We then put it in a drive and refuse to ever touch it because no one wants to do that, right? And so that's where I'm coming in and trying to bridge the gap a little bit and to automate some of what we're doing. And so this is what our current workflow looks like a little bit. We're going to fly the drone on a field. These fields are going to vary in size. They can be anywhere from under an acre to up to 20 acres, right? And so you could think if you fly a drone over a 20-acre field, you're going to collect a couple thousand images. We'll take those images and then use commercial software like Pix4D, OpenDroneMap, or Metashape, and stitch those into a unified orthomosaic. Along with that, we'll get a digital elevation map, which is super helpful and contains a lot of information about the height of the different plant varieties. But then we're faced with this really big challenge of, okay, now I have one giant image. I mean, these orthomosaics could be up to 20 gigabytes. And I'm only interested in specific plants within that image. And this is the part that's really limiting us right now. So currently we do a lot of this by hand, which means you just sit there and you have this big orthomosaic and you hand draw rectangles around plants. It is something that I would argue you could give a monkey enough coffee and they could probably figure out how to do it, right? It's not very mentally challenging, and it seems like something that computer vision should be able to automate. And so just to give us a little background of what we're looking at, this is an example of a cornfield. And so what we're interested in specifically is this one row. In that case, I have it over there in red. We're going to call that a plot. So that is a unique genetic instance. So every one of these is a unique set of genetics, right? It's a different one of those progeny that we created to start. And so we need to collect data on every one of those individual plots. Some other terminology, we call these things going horizontal ranges, and then we call the lines going vertically rows, right? So if I say row or range, that's what I'm referring to. And so with the help of Nathan Miller, we were able to come up with a mathematical based solution that uses a sliding window Fourier transform. And so when we look back at this image, we see that there's a lot of periodicity in the spacing of these plots. And that's by design, right? So when we planted these, we used a planter that was pulled behind a tractor. That planter typically will have either 4 or 2 rows. So when we drove the tractor, we planted these 4 or 2 rows perfectly spaced from each other. But what happens is when we turn the tractor around, when we made another pass going this way, we didn't drive in exactly parallel lines that we started with, right? So every 2 to 4 should be relatively consistent in their spacing, but then between those two, you're going to have a little bit of wobble. And so the approach that we came up with, we first just sample a sparse grid across the image to find the frequency of that planter that was used. Once we find that frequency, we lock onto it and we allow the phase to wiggle. And we then sample this image depending on its a parameter you can set, how much you want to skip. The more that you're going to sample this image, the more refined this, I'm going to call it a wave pad, is going to be. And so we can get a picture of this image in the frequency domain, and we see that it's very clearly picking up on those patterns of where the plots are. We can do this in the row direction or the vertical direction and the range direction or the horizontal direction as well. And doing that allows us to form a grid that we can then take the plots out of. And so this works pretty well in the middle of the field. The problem is on the edges of the field, because we're doing a sliding window, we can't sample the edges of the image. So what happens is you end up with this blank space that you have to then interpolate. When you do that interpolation, it ends up getting off a little bit on the edges. So it's not perfect, but what's really cool is that overall, it's the solution that we've come to is more right than wrong. And so that's to say that we could optimize this solution. And that's what we implemented in the next step. So what it does is scans across every plot that we've extracted and averages the pixel values for those to create a model. right? So we can see here, this is a first iteration model. It's not super great, but it has kind of a faint line of green down the middle. We can then take this model and optimize the center X, the center Y, and the theta of every plot. Once we perform this over all plots in the image, we can then recompute our model and then do another round of the optimization, recompute the model. And so in this way, we're stepping toward a stable solution. And we find that this works relatively well for these nice, clean images. So this is an example of kind of right in the middle of the season. There's not too much weed pressure. There hasn't been a windstorm. The plants are still standing up straight. And it works really, really well. The problem is they don't always look like this. And so some of the biggest challenges that we face are creating consistent grayscale images across growing season and crop type. So I've shown just examples with corn, but the goal is to have this work across potato to rice to wheat to all of these different people on campus are doing the same thing. And we're all faced with the same problem. So it seems like there should be a model that can pick up on all of this. The other biggest challenge that I've been running into is the computation time of the final optimization. Because we're doing that optimization and recomputing the model, then another optimization, it ends up taking close to an hour, currently the way that it's set up. And I think a lot of that has to do with the optimizers that I'm using. And so it's something that I was really looking forward to asking y'all questions on if you think there's any better methods. And so just to give you an idea of the challenges of trying to find the plants, it seems super simple, right? You should be able to just like look at the green stuff. And very early in the season, it is, right? So this is right after planting when the plants have just emerged a couple weeks. They're a couple weeks old. And right when they come out the ground, everything is going to be, the soil is brown because we just sprayed an herbicide. There's no weeds or anything. The plants are bright green. They're super easy to pick up. A little bit later in the season, the one that I had shown y'all, the plants are going to be bigger. They're more of a darker green. They're starting to crowd in on each other. Later in the season, you'll end up with all kinds of weed pressure in the field. So now, okay, we can't just find the green things, right? But clearly you can see that there's like a row crop and then there's weeds. And then towards the end of the season, you end up with the plants being entirely brown / kind of a yellow color. And this is a fun example I threw in, but again, we don't really know what we're doing. So this entire field was collected with the camera not set to autofocus, right? So the entire field is blurry. And then they just hand me an image and they're like, we'll get data out of it, right? And so there's a lot of just logistical problems with our data. There's now really standard operating procedures across all these public institutes. And so our pictures are all different sizes. They have different resolutions. And the plants look very different in all of these things. And so this has been really one of the biggest challenges. The other thing with the optimization is that it's not a continuous objective function. And so it's because we're sampling the pixels in this image, right? So you can imagine if I move the center X position by 0.0000001, well, I haven't actually sampled any different pixels, right? And so the objective function will do this, and then it'll do that, and then this, and then that. It'll kind of just wander all over the place. There's a ton of local minimums as well. And so this is kind of where I wanted to kick the can to y'all. Part of me thinks that this is an integer optimization kind of thing because I'm really only interested in moving by whole pixel values. But there's also that degree of rotation as well, right? So if I move in the X by half a pixel and then rotate by 10 degrees, now I can sample different pixels, right? So it's not sure if it's integer optimization or if it's just something close to it. And so currently I've been using particle swarm, genetic algorithm, and simulated healing, more stochastic optimization methods, and they work okay. They're just pretty computationally intensive. And so if there's any better way to do this, I would love to hear y'all's suggestions. And so the next step is through talking with Chris, he suggested trying to develop some kind of convolutional neural network to do this. Seems like it would be great for it. And I agree with him. That's an awesome idea. The problem we're at is we don't have enough training data. And so my goal in life is not to be the grand overlord of drone image processing. I'm tired of people just throwing me data and asking for results. So I've been working on developing a QGIS toolbox that you can use to implement the solution that we came up with, the sliding window FFT thing. So people can at least start to process their data now. the hopes is that we can then kind of create some community data set where we'll load in our orthomosaics and our extracted plots, which again might not be perfect, but in QGIS it has this neat feature where you can go in and manually edit each individual shapefile. So the idea is that would be the ground truth data, and then we could use that to try to train some kind of neural network. And the last thing here. So because this is so computationally intensive, I went ahead and developed a Docker container to run this on CHTC Condor. So the biggest thing here is setting up the param file. So you can kind of play around with this in QGIS to see what parameters are going to work for your specific image. Once you have that known, then you can just pass a parameter file to the Docker container, and it can just run everything. So you can throw it on some kind of node or cluster environment. So with that, I'd be happy to take any questions that you have.
Plant question, I guess. Two questions. The first one's a plant question. So for like corn, is there a good correlation between like how big the plant grows versus like how many kernels of corn get out of that? Or is that like how do you determine that just by looking at how the plant's growing?
Totally. Great question. So the answer is it kind of depends. So in corn, there's two main markets really. So one, the big one in Wisconsin here is going to be silage. So that's what you take your corn, you chop up the entire plant, including the cob, the stem, the leaves, everything. You then put that into a silo, you let it ferment, and you can feed it to cows. So in that case, you're really more interested in just growing tons of biomass. You're not really interested in the kernel size. In production agriculture, like if you just think of typical farmer Bob is just farming corn and we're just interested in the grain, there's not a super great correlation between biomass and kernel fill, because the more energy the plant's putting toward biomass, the less energy it has toward filling the grain. And logistically, it's just the bigger the plant becomes, the more likely it is to be knocked over by the wind. And plants that are knocked over by the wind can't be harvested, right? So there's a lot of different considerations.
All right, second question. So for your sort of non-AI based like plot segmentation, it seems like you maybe have already a lot of training data that you could harvest from that data that you could put into a CNN. Like, do you have the corners of each box in a convenient format?
So right now I have them in a GeoJSON format.
Okay.
So yeah, definitely workable. And I mean, we have like 25 terabytes of core data, but the problem I'm running into is just, it's so incredibly messy. And it's like, I'm going to spend more time just cleaning up this data set, because it's one of those, it's from all these different institutions. And essentially I'm just going to have to go into the drive and strip the metadata off of every image to figure out what actually goes with what, because there's no naming convention.
Yeah.
But if I can do that and then stitch all those images, then yeah, there would be a sizable data set to work with.
Because it seems that's basically like you need just the bounding box and the image to put into a lot of these models. There's we were talking about it earlier today. It's called YOLO, and this pretty popular CNN model, and there's pretty easy access to get quite far. There's a lot of tutorials on how to adapt it to your own data. You definitely want to check that out. I think it'll get you pretty far pretty quick.
Awesome. Yeah, definitely look into it.
Yeah, I think it could do plant detection right off the bat. It's just not going to be as great from the start as you want it to be. But if you have a small sample, you could fine tune YOLO and probably get some pretty decent results.
So is it going to give back like a bounding box, or is it going to give back like the plant pixels?
It gives about, you get a bounding box. They have to interact.
Yeah, I definitely got to look into that.
You mentioned that the original way that this is done is somebody walking down through the field and making measurements, which seems like a little bit different data than what you're getting from the aerial photos. I'm wondering, is there additional things that you would need once you've come up with the method of identifying the particular unique genetic phenotypes or plots? that you need to go to next to be able to distinguish between them and characterize them?
Yeah, so we have ground truth data on like flowering time, plant height, and ear height as well, are the three big traits that we're interested in for corn. So all that ground truth data exists with these image data sets, and we could use that to train because, I mean, you're totally right. It's like a totally different angle. So, yeah.
So is that collected by longitudinal collections, aerial collections of the drones at different time points through the growing season?
So, yeah, so we fly the drone probably 12 to 15 times throughout the season. And so the different flights correspond to different traits. And then we have like human collected ground truth data for all those traits as well.
And so I just taking on that, like, yeah, when you talked about this, you're like, oh, people are going out in the fields and measuring. Like when we look at your images here, they seem to be very far out. Like, are you actually, like, do you have more zoomed in, like, detailed images so that you can actually be like, yeah, from this drone, based on all the different views I posited, I can say that this leaf is 8.8 inches long, stuff. Like, I mean, it sounds like that's what's not there is, like, when somebody measures it by hand, you know exactly what it is. I feel like it's tough from those images to get those same types of measurements.
Totally. You're spot on. And that's what I've been arguing with my advisor. And it comes down to more of a logistics thing because like we have to fly these drones during the summer. And we're still collecting data manually while flying the drone. So there's only so much time in the day. So we end up flying these drones really high to make the flights go faster. But when you fly it that high, the resolution's so low, like you couldn't get leaf angle off the images I just showed you. Right. All we could really hope to get from the data that we have now is biomass from the digital elevation maps and maybe flowering time. So when the plant flowers, like the tassel, the thing on top of the corn plant turns yellow, which has a bunch of pollen. That you could see in these images, but like single leaves, not at all. I mean, the whole plant is like maybe 100 pixels in some of these images. So yeah, and that's something that an idea that I've been kicking around working with is the limitation is really on these DJI drones that we use. So for some reason, they only take a picture every 2 seconds. I don't know why. I mean, they're capable of flying at like 40 miles an hour and they have 8K cameras or 4K cameras, but they can only take a picture every 2 seconds. And so I've been playing with flying the drone really low and really fast and then just collecting 4K video and then just pull frames from that video and you can stitch images that are way higher resolution. And logistically, it's a lot faster to fly. So I'm trying to convince people to move towards that, but all the data that we have historically is this low resolution stuff.
Fly two drones too and get some stereoscopic vision out of it.
Yeah.
Fly high.
If you know anyone that knows how to like coordinate drones, we're getting contact with
People in ME.
Yeah. Well, I want to talk to the people. Whoever does like the drone show at the football games, I feel like that's the guys to talk to. I mean, it's the same thing, but with plants.
So I'm confused as to why segmentation is a little off when it comes to water fields. Like you said, you have to apply information, you know which the segments are, and then off. But if you start from the end where the cornfields are, instead of the outside, you technically be segmenting like any other image and stop at the last plot, for example, then it will still be aligned in that picture.
Yeah, so it comes down to when I use the sliding window for the sliding window FFT, that window has like a width and a height to extract the FFT signal from. And so like currently I set that window to like 1,000 by 1,600. Right. So then I can only put that window at the 501st pixel because it's going to extend 500 this way. And so like the one way to get around that, I suppose, would be to just zero pad the left side of the image and then you could sample all the way, but then you end up losing the FFT signal. So that's why we can't actually like find the signal on the very edges.
A clarifying question. you recap, how are you using the genetic algorithm? I kind of missed that. That's part of your optimization.
Yeah, so
To take care with the FFT parameters, or
it's to optimize, where are we at? So it's to optimize this placement. So I take every one of these boxes and then I move it to the left, to the right, up, down, and rotate it within like a certain range of parameter spaces. And then that's what I'm using the genetic algorithm to.
what's the error that you get? How do you measure the error?
From this model. So where is it? Nope, up there. From this model. So then the new image that you create, you just subtract from this and take the norm of that image.
Okay. Gotcha.
So the idea is as it gets closer to this model, that it's
Genetic algorithm definitely brute force. You get good results, but they're compute intensive.
yeah, I mean if you just let it run for like an hour and a half, it works great. But it's like I'd rather not have to let it run for an hour and a half. But maybe that's just part of the game. Awesome. Well, thank you all again. I appreciate the opportunity.